Okay, how's it going?
It's going good.
And so what are you doing here in the Amazon?
Well, this trip is to install equipment to measure solar-induced fluorescence from the canopy. So we've been installing a fiber optic that goes to the top of the canopy, a sensor that looking down and looking up from a perch above the canopy, and then analytical equipment that operates down here at the ground to estimate or calculate the fluorescence that's coming off the canopy.
And so why are you trying to measure solar-induced fluorescence?
Well, fluorescence is sort of an inevitable part of plants doing photosynthesis, and it's directly coupled with the amount of light that they intercept and the efficiency with which that light's being used. It's been a very major part of photosynthesis research for years. There's last time I looked, there were something like 30,000 papers on Google
Wow.
if you search for chlorophyll fluorescence. So it's a major research tool used to examine mechanisms of photosynthesis. And the really cool thing that we've got is in the last 5 years, we've figured out how to get fluorescence measurements with satellites from space.
But most of those fluorescence measurements have been done like at the leaf level, right?
All the stuff those 20 or 30,000 papers are leaf or pieces of leaves or algae, really small scale stuff.
And so now the sensor you've installed here at the tower what scale is that picking up the?
Well, it's looking at probably 50 meters diameter patch of forest. It looks it's a full hemispheric view, but it's a cosine weighted view. So the most of the light that we're collecting is from the say 30-degree cone.
Okay. And what's some of the major challenges in terms of like the scaling of fluorescence from the satellite to the leaf level?
The biggest challenge is that the satellites that we're using look at really big patches, like 30 or 40 kilometers on a side. And what you'd like to be able to do is to say, okay, how much photosynthesis was going on in the piece of ground or the pixel that I've looked at.
Mm-hmm.
And it's pretty hard to do that at the scale of 30 or 40 kilometers. So what we're trying to do is to do this at an intermediate scale, and we'll be doing some measurements of fluorescence of the leaves. So we're scaling from the leaves up to the tower, and then it's a big jump, but we think that that's an important. We're seeing the leaves then under their natural conditions.
Mm-hmm.
So it'd be a way that we can better calibrate what we are seeing from the satellite and better interpret what the measurements really mean.
So one of the goals is to be able to measure photosynthesis from the satellite.
Yeah, I mean, one of the things that's been a persistent problem is that we understand photosynthesis extremely well at the scale of the lichar.
Mm-hmm.
But as we get to bigger scales, even the tower, we make measurements, but they are, if you average long enough, you get good measurements, but you can't get as good a measurements from a tower as you can from a lichar. And once you get beyond the tower, we really have no mechanism for understanding what the rate of photosynthesis is at larger, sort of geographically useful scales. And since we know we've got this fluorescence signal, and that it's closely tied with the mechanisms of photosynthesis that we understand pretty well,
Mm-hmm.
we can then make a run at seeing, well, what use can we make of this? And that's really what we're doing is, we have a few ideas of how it might be useful, but basically it's a suck and see kind of thing. What can we get out of it? It's worth looking at. It's new.
Cool.
It's exciting.
And how long have you been working on photosynthesis? You kind of
Well,
were
I did
involved
my
in some of
PhD
the...
on photosynthesis, so I
Okay.
started that in 1966.
Oh, wow.
So 50 years ago?
Were you working with like gas exchange equipment, or what was your
I was
project?
a biochemist at the time, so I was working
Okay.
on a little bit with gas exchange, but what I worked on was this process called C4 photosynthesis.
Mm-hmm.
So I was looking at the biochemistry of that and was involved in figuring out the role of compartmentation of the reactions in C4 photosynthesis. And then I sort of have been moving up in scale ever since then. And of course that was a move up in scale. I was a physical chemist to begin with. As an undergraduate, I did a lot of physics and chemistry and was involved in sort of the early NMR and EPR and stuff like that. And then thought, oh geez, that wasn't making sense to me. Maybe something more coupled with nature would make sense. And as I got into nature, it started at small scales and kept getting bigger and bigger. So my goal now is to figure out some way to put together a measurement of photosynthesis by the planet.
And you're also working with carbonyl sulfide, is that right?
Yeah, carbonyl sulfide is an interesting gas in that it has a sulfur in place of an oxygen in carbon dioxide. So it's an analog of carbon dioxide, which is the gas that's used in photosynthesis, and behaves very much in terms of in photosynthesis like carbon dioxide. But it's got a useful property that we see when we look in the atmosphere. If you look at the atmosphere over North America, for example, the carbon dioxide may change seasonally by 10%, and carbonyl sulfide changes seasonally by 50%.
Oh, wow.
And they do so in synchrony.
Hmm.
So that says there's something happening to carbonyl sulfide that's not happening with carbon dioxide. And what that is that carbonyl sulfide doesn't have very much in the way of sort of little internal cycles going on over the continent. Over the continent, CO2 is being produced here, and it's being taken up there, or it's being produced in the day here and taken up at night, or it's being produced at night and taken up during the day.
Mm-hmm.
So it's kind of going around in a whole bunch of little cycles, and you can't really see what's happening with it because it's kind of short-circuiting your measurements at larger scales.
Mm-hmm.
But carbonyl sulfide doesn't have those short circuits. So the idea is that we'll be able to measure it, for example, here over the Amazon, and we'll see it going down at a rate that's proportional to the rate of photosynthesis, which is linked also to our study of fluorescence.
Okay. So
hopefully the idea here is that we would be able to get a better idea of what the total photosynthesis is of the Amazon region.
So it's affected by photosynthesis, but not necessarily by respiration, or...
That's right. The cycle of photosynthesis is between respiration and photosynthesis, and they co-occur, basically.
Mm-hmm.
And the cycle in the atmosphere of carbonyl sulfide is it's produced over in the ocean by the action of UV light on organic matter in the ocean. And it comes up in the atmosphere over the ocean, spreads out across the top of the atmosphere, and sort of mixes down, and it's being consumed over the continents by the interaction with plants.
Cool. And you were just recently selected or elected into the U.S. National Academy of Sciences.
Yeah, it's kind of a strange feeling. I still haven't quite assimilated it. I think of this as I got this friend who works in science, and
Uh-huh.
I have he's famous now, and I have to take care of him. But he hasn't changed me.
That's cool. So what are some of the things that are required to get into such a prestigious academy?
Well, I don't know. I think it's part of it's having friends in high places.
Yeah.
Part of it is having written a few good papers and kind of pushed the field along.
Mm-hmm.
I don't know. There are so many really good people I know who aren't in the National Academy.
Mm.
And I can't think of a reason why I'm there and they're not.
Mm-hmm. Yeah. But you were involved in a lot of sort of fundamental papers on photosynthesis, right? Like describing models that would predict photosynthesis based on stomatal conductance and gas exchange.
Yeah. I think it's a matter of having been pretty lucky in the sense that things that I worked on turned out to be useful.
Okay.
I mean, I had no idea when we were doing them that they'd be useful.
Yeah.
They were just interesting.
Okay. So is that sort of your philosophy in science, to just do things that you find are yourself personally interesting? And
well, yeah, personally interesting, and also I think, like everything else, I guess I think of science as being kind of an art.
yeah.
That you look for something that you can create something that you think might be beautiful out of.
Mm-hmm.
And to make to understand how something that wasn't well understood before, understand how that works.
Mm-hmm.
Put together fields or experimental measurements that haven't been put together before, or to organize it all into a simpler equation. All that kind of stuff is kind of fun when you can identify it. And I think of myself as being kind of a vulture flying around looking for these opportunities to dip down and feast.
Nice. But some of the students here in Manaus have described you as just like a postdoc, because you're in there with the hands-on, with the equipment. Like, for example, you're here in the central Amazon installing a fluorescent system, which is not really, I guess, typical for someone at your stage in your career. Have you just made a sort of a big point to try to keep a hands-on in science this whole time, or?
Well, gee, I suppose, I mean, there are two things involved in that. I work for the Carnegie Institution, which provides me with support to do research. I don't have to worry about supporting my salary. They cover that. I can get grants, but I don't even have to get grants to continue research because they provide me with some support. So that is a really lucky situation where I can do that. I've also discovered that I'm not a good administrator.
Uh-huh.
And it would be far better with somebody who's more capable of reading the fine print and negotiations and things like that, dealing with the details of running an institution. My wife takes care of our finances. they shouldn't put me in charge of a large institution's finances. Uh, I, I've always considered that I'm just better at doing science, so why change?
Yeah.
Uh, and because I have the opportunity to do that, and because I had probably the most fun of my life when I was a graduate student,
Yeah.
why not just keep trying to have fun at it?
Oh, that's a good way to do it. And what do you think in the next century or so forth is, these predictions of global warming and perhaps, Amazon collapse even? What do you think, what's my question here? I guess, what do you think is the future of photosynthesis research in terms of, like, in the context of climate change?
Yeah, well, I guess the thing I'm looking at here is that the planet really runs on photosynthesis. It's the energy supply for the biosphere. And, climate change could happen and photosynthesis and the biosphere could keep right on trucking. And it might cause us inconveniences, but it wouldn't be a serious problem. But if photosynthesis or other key processes that essentially run the planet start going south on us, then that's a real problem. And right now we would have no way of knowing